Welcome to this ACUR at ANU student presentation session, everyone. I'd firstly like to acknowledge and celebrate the first Australians on whose traditional lands we meet and pay our respect to the elders past and present. My name is Max Kirkby and I will be chairing today's session. So our next presenter is Neha Bal. Neha, please feel free to get started whenever you're ready.
Thanks, Max. Right. Good morning, everyone. My name is Neha, and today I'll be discussing the metabolic consequences of obstructive sleep apnea on the liver and how this might drive cardiometabolic disease. So obstructive sleep apnea, or OSA, is a chronic respiratory disorder, and it affects about 1 billion people around the world. So patients with OSA experience a repetitive collapse of the upper airway, which leads to pauses in breathing during sleep. Concerningly, OSA patients are about 25% more likely to have type 2 diabetes and about 10% more likely to have cardiovascular disease than the normal population. And these cardiometabolic diseases also have a great economic burden, and they're predicted to cost about $58 billion annually by 2031. But despite this, we don't really know how OSA might be causing cardiometabolic disease. So OSA is characterized by a number of physiological changes. It can cause pressure changes in the chest, sympathetic nervous activation, hypertension, and intermittent hypoxia. So this is an oxyhemoglobin trace from an OSA patient, which shows how these obstructive apneas throughout the night can cause periods of hypoxia or low blood oxygen, followed by reoxygenation. And studies in animals and humans over the past decade suggest that intermittent hypoxia might be a key driver of the cardiometabolic dysfunction that we see in OSA patients. Clinical studies show us that OSA patients have a higher incidence of metabolic disorders like hypertension, insulin resistance, and hyperglycemia. And they've also found a higher incidence of these disorders with increasing hypoxic events per hour in OSA patients. So together, these findings show us that intermittent hypoxia might play a key role in the development of cardiometabolic comorbidities in OSA patients, but it's difficult to isolate the independent effects of intermittent hypoxia from confounders such as obesity in human studies. So animal models allow us to study intermittent hypoxia without confounders such as obesity. There are many animal models that have been used, but the most commonly used model, and the one that we used in our study, uses hypoxic gas mixtures to simulate fluctuating blood oxygen saturations seen in OSA patients in rodents. We know from human studies that OSA causes fluctuations in blood oxygen, leading to intermittent hypoxia. And as shown by these traces highlighted in green, we also know that our model of intermittent hypoxia causes blood oxygen saturation fluctuations in rodents. We can measure oxygen fluctuations in tissues using oxygen electrodes, which can't be done in humans because it's highly invasive. So one pivotal study measured oxygen fluctuations in lean mice, and they showed, as highlighted in the blue traces, that when compared to muscle and fat, the liver showed the largest swings in oxygenation. Findings from many other studies also show us that fat and glucose metabolism in the liver in particular might be dysregulated by intermittent hypoxia. So considering these findings, we think that intermittent hypoxia might alter metabolism in the liver, and this might be the key driver in the development of cardiometabolic comorbidities in OSA patients. So although these metabolic changes in the liver have been reported in many rodent studies, there are often conflicting changes reported across different studies which use different intermittent hypoxia exposure duration. So it's difficult to pinpoint whether these differences are because of a difference in exposure duration or because of a difference in experimental conditions across different studies. And understanding how intermittent hypoxia and OSA might affect metabolism over time is really important because many OSA patients are unaware if they've recently developed OSA or they've been living with it for years or decades before their diagnosis. So we're attempting to address this gap in the literature by investigating into how intermittent hypoxia might alter metabolism in the liver over time, by assessing changes in key metabolites and enzymes in response to both short and long-term exposure to intermittent hypoxia. So to understand how altered metabolism in the liver might drive cardiometabolic disease, we need to go back to the basics of how the liver functions on a cellular level. In the fed state, the liver uptakes about 2/3 of glucose from the bloodstream and breaks it down through a number of metabolic reactions to produce energy. And the excess products of these reactions are then stored in the form of glycogen and fats in the liver cell. They can also be delivered back to the bloodstream via lipoproteins. In the fasted state, less glucose is available in the blood. So liver cells can adapt to this by endogenously producing glucose or breaking down stored glycogen or fats. The liver can also produce ketones, which it can deliver through the component of blood known as plasma to the heart to provide energy. So this direct exchange of metabolites from the liver cells to the plasma is central in the regulation of metabolism in our bodies. So therefore, the second aim for our study was that we are looking at metabolite levels in the plasma and whether these changes correlate with those seen in the liver. So to investigate these aims, we're using the hypoxic gas rodent model that I described earlier to study the independent effects of both 8 hours of intermittent hypoxia or an acute exposure and 7 weeks of exposure to intermittent hypoxia or a chronic exposure, but all under consistent experimental conditions. So at the end of these acute and chronic exposures that we did earlier this year, we collected the liver's plasma and tissue from the rats and flash froze them. And we then used mass spectrometry to measure or quantify metabolites and Western blotting to detect protein levels and of enzymes in both our liver and plasma samples. So in the first stage of our investigation, we used liquid chromatography and mass spectrometry to measure metabolite levels in liver tissues from our acute and chronic rats. To separate and identify metabolites, a mobile phase carrying the sample mixture is passed over a stationary phase. So analytes in a mobile phase, which have a strong interaction with the stationary phase, will be bound for longer, whereas analytes with a poor interaction will elute out of the column earlier. And then the relative abundances of these metabolites can then be measured using mass spectrometry. So using this technique, we found that the ketone body acetoacetic acid was significantly increased in the livers of rats exposed to acute intermittent hypoxia, but remain unchanged in those exposed to chronic intermittent hypoxia. When blood glucose is low, the liver can produce ketone bodies in the form of acetoacetic acid and 3-hydroxybutyrate. And they can export these through the bloodstream as a source of energy for other organs. The slowest reaction, and therefore the rate-limiting reaction of this process, is catalyzed by the enzyme HMGCS2. So to further investigate the regulation of the ketogenic pathway, we then measured protein levels of the enzyme HMGCS2 using Western blotting. Proteins can be separated based on their size through gel electrophoresis. And then these separated proteins can be labeled with a luminescent substrate that can be visualized using an imager. So the darker the band on the blot, the more protein has been detected. So through this method, we found that HMGCS protein levels were significantly increased in the livers of rats exposed to acute intermittent hypoxia, but they remain unchanged in those exposed to chronic intermittent hypoxia. And these results support the changes we detected in the ketone body acetoacetic acid through our metabolomics screen. Ultimately, we think that intermittent hypoxia alters metabolism in the liver, but that there might be adaptive or maladaptive responses which are activated in the short or long term. after exposure to intermittent hypoxia. And this might affect energy substrates for other organs such as the heart. And we think that this is the link to cardiometabolic disease. So going forward, we'll also be looking at changes in the plasma metabolome and how changes in the liver might be reflected in the plasma. And understanding these alterations might be a pathway for targeted drug discovery or early detection and prevention of cardiometabolic diseases in OSA patients. Thanks everyone for listening, and thanks to my supervisors and group members at the HRI and CPC. Thank you.
Thanks very much, Neha. There are now 5 minutes for questions and Q&A. Judges, do you have any questions to begin with?
Thanks, Neha. That was really interesting. I was wondering if you could talk a little bit more about the difference between the acute and the chronic exposure and what the implications of that are. So it's kind of how to make sense of the fact that acute matters and chronic doesn't in your research. Because I would have expected, I guess, that chronic would have had more of an impact, considering that the human model. Yeah, so what does that imply, do you think, and in terms of next steps?
Yeah, so we think that there's an initial, the initial metabolic response to acute intermittent hypoxia is kind of like a stress response. And so we see more drastic metabolic changes. And although there might be differences in what we see in the physiology and the metabolic responses, eventually we need to look more in the metabolic responses because these can be hidden under the surface and you can't really tell sometimes in the physiology what's happening inside, and then that can have longer term implications. So with our chronic exposure, we see a more dampened response, but this is the longest exposure compared to other literature. They usually do about 3 to 4 weeks. So we're not even really sure if this really replicates the maladaptive stage of the disease. So what we think is that there's this initial maladaptive response where we see huge metabolic changes. And then somewhere in the middle, maybe 4 to 6 weeks to 7 weeks, there's some type of adaptation. And that's why we might see in OSA patients who are in that stage of the disease, we might not see a lot of metabolic changes, but then further on following maybe at 12 weeks or something, we might see maladaptation again. So we need to really understand this change from adaptation to maladaptation so that we can predict maybe down the line how metabolic changes in the OSA patient might have worse outcomes. And then if we can pick that up earlier, then we can take preventative measures.
That's great, thank you.
Are there any questions from the audience? And if so, please type your question into the chat or raise your hand so that I can unmute you. So it doesn't seem like there are any questions from the audience. And so, yeah, I think that's it. Thanks so much for that fantastic presentation, Neha. It was really insightful. And altogether, that brings us to the end of today's session. So thank you to our judge, Dr. Celia Harris, and congratulations to both Edward and Neha. You've done a fantastic job. So this recording will be made available online following the conference, and we hope to see all of you in other conference sessions throughout the remainder of this week. So thank you very much.
Well done, everyone. Really nice presentations and really interesting work. So congratulations.